My name is Janay. I'm a PhD student at McGill, and I'm going to be talking about the impacts of predation stress on DNA methylation and behavior in guppies. So we know that early environment can have long-lasting impacts on behavior, and these impacts can be adaptive or maladaptive. If the early life environment matches the environment experienced later in life, then behavior can be adaptively primed to match this environment, or sometimes developmental shifts in behavior can be maladaptive responses to stressful environments. A variety of environmental cues can induce these shifts in behavior, but predation pressure is a particularly strong cue, with increases in predation typically resulting in increases in anti-predator behaviors or decreases in risky behaviors. So for example, in this little schematic here, we have a juvenile exposed to predation stress during its early life. It grows up and then exhibits a changed alarm response as an adult. Epigenetics have recently emerged as a potential underlying molecular mechanism for phenotypic plasticity, especially environmentally induced plasticity, and DNA methylation is the most well-studied of these mechanisms so far. This is the addition of a methyl group on a cytosine, often in a CpG dinucleotide, which is just a cytosine and a guanine next to each other, and it plays an important role in gene expression, with increases in methylation typically associated with decreases in gene expression, although that is not always the case, and it can sometimes also result in increases or alternative splicing patterns. We also know from a bunch of emerging studies that early life environment can have stable impacts on DNA methylation that last well into adulthood, which suggests that it could be a mechanism of developmental plasticity. And accordingly, a bunch of studies have come out suggesting that methylation could play a role in behavioral plasticity, with a lot of these studies focusing on the impact of early life stress on behavior later in life. However, most of these studies focus on mammalian systems so far. A lot of them are in mice or rats, and they often try to recreate environmental conditions that are relevant for human research, such as maternal care or other types of relevant stressors. So we don't really know how relevant methylation may be for behavior in wild populations so far. This is where my study system comes in. So Trinidadian guppies are small tropical fish. They live in streams like the one seen here, where there's a waterfall barrier. The guppies live on both sides of the waterfall, but their predators only live on one side, which separates them into these nice low predation, high predation dichotomies that have been the subject of a lot of research over the year, establishing them as a pretty big evolutionary model system. A lot of the research focuses on their coloration patterns. As you can see, there's a lot of variety in male coloration from these pictures here. But there's also a lot of work on their behavior, with predation typically being associated with increases in neophobia, increased boldness, and increased shoaling. Guppies also react strongly to alarm cue, which is a reliable predation cue that is released from the skin during mechanical damage, such as during a predation event. And conveniently, it can be produced easily in the lab to induce predation stress. When you add alarm cue to a tank, the guppies lower in the water column, they avoid areas with the cue, they freeze, and they start dashing around the tank. Guppies also show developmental plasticity in response to these predator cues, with guppies exposed to predator cues showing increased shoaling under acute predation stress, increased risk sensitivity, cognitive flexibility, and larger brains in males only, though. So my hypotheses for this project were first that early life exposure to alarm cue would induce developmental shifts in behavior in guppies, and secondly, that methylation could potentially underlie this developmental behavioral plasticity. So for my predictions, if we go back to how I said that predation impacts behavior, then we would predict that alarm cue exposed fish would be less exploratory, because exploration is moving around a novel environment, and being neophobic is being afraid of novel things. They would be more bold, and they would shoal more. For my methylation predictions, I predicted that there would be differential methylation between the alarm cue and the control fish, and that this methylation would be located in genes that are involved in behavior. You could also expect that there might be sex differences in methylation responses in both the magnitude and which genes are impacted. This could go either way. So the females are known to respond with stronger anti-predator behaviors than males. So you could expect that females might have a larger magnitude in response than males. But males are also known to adjust a wider array of behaviors, including mating behavior in response to alarm cue. So potentially maybe males could have a higher magnitude of response as well. And then if methylation is an underlying mechanism of behavior, developmental behavioral plasticity, then you would expect that there would be associations between methylation and the behaviors that are impacted by the alarm cue. So for my methods, I used Paria origin guppies, which are from a low predation habitat. I gathered juveniles that were born within 1 week of each other, separated them into tanks, and then exposed them to either alarm Q or control Q, which was just water, 3 times a week for 8 weeks. I then separated them into sex-specific housing and allowed them to mature to adulthood for 22 weeks. And this time without Q allowed me to disentangle the differences between a developmental exposure and a recent experience. Then I put them through 2 behavioral assays, extracted brains, did whole genome bisulfite sequencing to analyze data for differentially methylated sites and regions. The behavioral assays I did were an open field test where the fish was allowed to explore the tank and there was a plant in the corner. And from this I could extract exploration, which was the unique squares explored, and boldness, which was the time that the fish spent either hiding in the plant shelter or along the outer edge of the tank. And I also did a shoaling test, and in this test, I introduced 2 containers into the tank. One contained a shoal of 4 females, and one was empty. And from this, I could measure sociability, so the fish's preference for the container with the shoal. For my results, first, what I found was that alarm Q exposed fish were not bolder or less exploratory. So if you look in these figures here, the different behaviors are on the y-axis, and then alarm Qs on the left and control Qs on the right. As you can see, there's not a major shift in behavior. This could be for a couple of reasons. The first is that there's conflicting fitness trade-offs for these behaviors. So being more bold or more exploratory in a high predation environment would expose you to more risks, but it would also expose you to more opportunities. And also, it's just possible that these behaviors are not developmentally plastic in this population, which doesn't mean that they're not plastic in all guppy populations, since plasticity can evolve. And so sometimes we see that in high predation populations, they're more plastic than low predation. But what I did find was that alarm Q exposed fish shoaled more. And the reason that we think that this behavior in particular was sensitive to the alarm Q exposure is that there's a pretty strong benefit of shoaling under high predation, where being in a shoal protects you from predators. For my methylation results, I found significantly differentially methylated sites and regions for both males and females. But if you look at the numbers, there's clearly a lot more for males than females. And also, there's pretty low overlap between the sexes. So this suggests that males could be more responsive to a developmental alarm Q exposure, at least in terms of their methylation responses. And as I previously mentioned, this could be because of the fact that they also shift their mating behavior in response to alarm Q. And this shift in mating behavior is thought to be more cognitively demanding and is also associated with shifts in brain size and morphology. Also, the low overlap in DMSs and DMRs between the sexes suggests that there's different genes involved and that methylation is likely playing a different role between the sexes. Then I wanted to associate my methylation with behavior, so I focused on shoaling behavior, as that was the only behavior that was significantly impacted by Q, and I looked at the individual differences in methylation at the DMRs. To do this, I used an elastic net model, since basically my outcome variable was shoaling, and then I had many predictor variables, which was every DMR. An elastic net model basically uses penalties to perform a type of feature selection where some of the regression coefficients of the different predictors are reduced to 0, therefore retaining only the DMRs that actually covary with the behavior. What I found was that models had low predictive power overall, suggesting that methylation at all the DMRs is not predictive of shoaling. But when I focused on only the important DMRs that were retained in the model, I found that some specific DMRs were significantly associated with shoaling, for males only though, although one of them approached significance for the females. So in summary, what I found was that alarm Q exposed fish were not more bold or less exploratory, but they did shoal more. And then for methylation, I found that there was differential methylation between the alarm Q and the control Q fish. There were also sex differences where males showed stronger responses and in different genes, suggesting a different role of methylation between the sexes. And I did find associations between methylation and behavior. However, these were significant for the males only. So in conclusion, I found that an alarm Q exposure during early life induces stable shifts in both behavior and methylation for guppies, suggesting that DNA methylation could potentially be a molecular mechanism of behavioral plasticity in guppies. But importantly, I found that there were major sex differences in the responses, which, as I suggested, could potentially be due to different behavioral responses, maybe shifts in mating behavior that females don't exhibit. But future studies could definitely investigate these shifts in mating behavior more. So with that, I want to acknowledge my supervisor, my committee, all my other collaborators, and the Barrett-Hendry Reader Lab members for all their help, and then my funders. And then, yeah, if you have any questions, you can also follow me on X. The paper should be coming out soon, or give me an email. Thank you. Yep.
Two questions. Do you know where in the sequence the DMRs were? Like, were they at introns, promoter regions, gene bodies? And second, have you or will you be trying to see if they're associated with either genes or structural variants or something other?
Yeah, so I did find an increase in DMRs in promoters and introns. I have that data for the paper, I just didn't present it, but in terms of structural variants, so do you mean like in gene
No, I'm just saying as an example of other non-Mendelian.
Yeah, so it would be nice. I think another student is now doing a follow-up project, doing a different type, I think he's doing chromatin stuff. And also I was initially supposed to do gene expression as well, but the brains are just so small that my RNA samples weren't very good, but I could maybe do some qPCR on specific genes that I know are differentially methylated to see how this methylation is impacting gene expression. Yeah.
So the females didn't show a lot of differentially methylated regions. Is that because the females in all the treatments, their methylation profile was the same, or there's just so much variation?
That's a good question. I should look into that. Yeah, I actually don't have an answer for that, but I can definitely check that. Yeah.
Kind of a technical question that you sort of hinted at in your last response, but just with the brain instructions for the self-isolation, did you modulate the whole brain, or did you choose a particular region? And so what did you choose?
Yeah, so I use whole brain for this project because of limitations and how small the brains are and getting enough DNA. But I would suggest that future studies could definitely try and do specific regions, although if they do that, they would likely have to pool between individuals. So it's kind of a trade-off on like whether you're getting this heterogeneity within the brain or between individuals. But I mean, maybe if somebody did maybe like single cell sequencing, I was initially supposed to use like laser capture microdissection and try and do that. But because of the pandemic, I couldn't do it. But yeah, so I used whole brains. Yeah?
Does color changes in animals relate to location, predation, or another factor? You mentioned that earlier.
Color?
Yeah.
Color changes, it's often studied in terms of predation, so it's like usually a decrease in coloration with high predation environments, but it's also pretty population specific and not as parallel as people say. It's also like heavily sexually selected in these fish.
One more quick one. Okay, thank you very much.
Great. Thank you so much to the organizers for this symposium, and it's great to see this turnout because I think there's a lot of energy and excitement about this topic, so it's really great to get together. So my lab, we study the, among other things, the Colorado potato beetle because it's a charismatic insect pest that has basically started the pesticide industry and has been a global invader. So it has made it around the entire world across all of the northern hemisphere. And definitely it was the way was paved by potatoes, but its ability to adapt to all sorts of novel kind of stressors and environments seems to be characteristic of its story. So I'm presenting work from many different students, and I have to say we are sort of in between data sets, so I'm sorry I'm going to present maybe a little older paper right now, but we're on the cusp of a lot of data. So agriculture is a really excellent way for studying rapid evolution. As many of you have heard, pesticide treadmill is a textbook example of evolution at work, where kind of you might have a population, and upon exposure to a pesticide, only some survive, and then that they propagate, and that helps to perpetuate the pesticide treadmill because of increased resistance. We also think that studying this pesticide treadmill is an excellent way for studying stress-induced evolutionary processes because agricultural pests are constantly targeted by a range of different kinds of stressors. And in addition to insecticides, there are fungicides, herbicides, high levels of toxicity, and other things like, say, natural enemies. And we know that kind of following sublethal exposure to insecticides, we see the survivors gain kind of tolerance to all sorts of different insecticides. And this has happened over and over again, and this is kind of the underlying kind of physiological kind of mechanisms that leads to resistance. But one of the things that we've been thinking about more is this idea of insecticides are another form of stress. They are sometimes modeled after naturally occurring compounds, sometimes not. And this is a particularly helpful paper for us in thinking about the range of mechanisms or responses that happen following stress, that you can have all sorts of different things that follow from that. Changes in mutation rates, changes in histone, post-translational modifications. There's chromoanagenesis, which is the shattering of chromosomes and the reassembly of them as well. And then also transposable element activity. So we posed first a thought paper. This is after being unsuccessful with grants, and we thought if we at least publish these ideas, we don't have to spend as much time in the preamble trying to justify the grant. And so we decided to just come out and just say, what if, with, all sorts of different kind of epigenetic modifications that they're inherited, and it's really some of these changes that are rapidly responding to following insecticide exposure, and that over time that it's partially, we're not saying that there's no more kind of genetic inheritance, but that there's also epigenetic inheritance as well that could help to explain the rapid onset of insecticide resistance. And this is work from my student Christian Breivik, who then went on to do an experimental evolution study where we took imidacloprid, this is the pesticide of the decade right now, it's a neonicotinoid, and we asked, well, how does the sublethal exposure to imidacloprid affect DNA methylation in both the target generation and subsequent generations? And we took what he called the high dose, but this is a dose that only killed 10% of the beetles. So it's actually sublethal. Anything under 50% is sublethal. We took 1/10 of that dose, so presumably this is less stressful, and this was considered an LD0 because nothing died. And then we also modified the compound itself to make it less toxic, and this also did not cause any mortality as well. And then we had a control of water. And we exposed the larvae in the parent generation, and then we wanted to look at what would happen in subsequent generations. So at this time, there was maybe a little less kind of studies published and stuff, and so we had a smaller pot of money, and so we were a little nervous. So even though kind of looking for kind of signs of differential methylation in the F3 generation would be really pretty cool and significant, I chickened out, and we went for the F2 generation because we only had a certain amount of money. And this is the PhD thesis we're talking about. So this is what other scientists have called the intergenerational effect, meaning kind of in this generation, something could be exposed as an egg, for instance, in a female, and so we don't know if it's truly kind of a transgenerational effect. And so what Christian found was that basically the sublethal exposure caused differences in global methylations. So this is CpG methylation across the parent and F2 generations. And this is following kind of whole genome bisulfite sequencing. And what was remarkable is just the patterning was consistent for both the exposed generation and the F2 generation. And these are beetles that are derived from a colony. There was really no reason that any beetle should be any different than the next beetle kind of thing. But we figured that taking kind of, maybe a mixed colony population and exposing to the treatment, we should see that the treatment effect overrides sort of any kind of genetic background. What Christian found was that, surprisingly, if you look at these, the three bubbles represent the three imidacloprid treatments. And you see that in the middle, I don't know if you can see that actually, it's only 13 sites that were shared across these three treatments. suggesting that there's a lot of sort of variation in stochasticity, even with the same sort of stressor, the same compound, base compound. So even though there was 221 differentially methylated sites, this was early on where the genome was not as resolved, for instance, and so only 9 of these sites fell in annotated regions. So there were cytochrome P450, and then putative cyclin-dependent kinase, which is important for cell cycle regulation. The interesting thing is that in terms of the sites where the differential methylation occurred, it was overrepresented in TE sites. And that was 39% of those total sites. And this is just a table where the differential methylation sites were, and these were kind of within 2 kb of these particular genes. And they were basically what I just told you, cytochrome P450s, and then the cyclin-dependent kinase. Okay, so one of the things we also have been wondering is kind of like, why is there potentially demethylation that comes? And so one of the things, as you know, is that for DNA methylation to occur, it's dependent on the carrier S-adenosylmethionine. So this is the carrier for the methyl group, which is right there in the middle, associated with the sulfur element. Methionine is actually one of the most limiting essential amino acids in plants, for instance. And what's interesting is it's all linked together with through the 1 carbon cycle, where you see glutathione, which is typically used to conjugate with toxins, is dependent upon also the availability of homocysteine. And then the top of it is kind of where SAM is generated as well. So it's possible that exposure to toxins leads to perhaps an increased expression of glutathione that draws down the availability of homocysteine that makes SAM actually more limiting, and then also might lead to hypomethylation. So, so at this point, I would just say kind of from this study, we see that sublethal exposure to insecticides alters DNA methylation, that selection for resistance can lead to transcriptional and methylation patterns. Some of that is from another student that I'm not sharing with you. We've seen substantial changes in physiological responses following that exposure as well. And so we see that at least can be passed on to 2 generations. So at this point, we are in the middle of an experiment to try to look at the transgenerational effect. And this is where we are actually, because our previous work was looking only at sort of this like colony mass rearing kind of approach, where we don't know who's the parent and what their background was. And here we did the opposite, where we basically are taking kind of a sexually breeding insect. And that's kind of the contrast to a lot of the other data sets that have been generated, is that we're taking kind of the parent generation, exposing them to insecticides, and then taking their progeny, and then subjecting them to multiple generations of incest, and then looking at the whole genome bisulfite sequencing, looking at the transcriptome, larval performance, and then we're also looking at histone modifications. So far, looking at both the F1 and F2 generations, we have not seen any changes in histone modifications that we know are responsive to the insecticide stress. So that part of it was a little bit of a disappointment, but we are still kind of collecting all the other components of the data. And so we've been kind of pairing up beetles, and this has been a massive undertaking where siblings basically are getting mated, they're reared in separate cages, we're sorting pupae, and we're sexing them, and we have multiple contingency pairs, and then we have kind of the next generation. At this point, we are just finishing up that F3 generation, so that's why, unfortunately, I don't have data to share with you right now, but you could see we've kind of expanded, so we have 12 lineages, and some of them didn't make it. But at this point, we've seen that there's variation among the lineages in terms of baseline tolerance. And so we will be able to sort of partition what is the role of kind of genetic inheritance versus epigenetic exposure. Yep, so far, this is kind of what we're aiming to do in terms of whole genome bisulfite sequencing. And at this point, I can close by just thanking my lab. We call ourselves the Insect Agroecology and Evolution Lab, and we are only 2 hours away. So if you're interested, you're welcome to stop by afterwards in Vermont. Yeah.
I have to always preface my questions when I hear about insects that I study plants. So I'm always trying to see to what extent my impressions of methylation apply to insects. So I'll ask a few. One is the TE methylation overlaps with the methylation. Where were you seeing that? Was that in promoter regions and was predominantly with CHH methylation?
It's been predominantly CPG methylation, and overall in insects, the methylation levels, and actually among orders, it varies in terms of how much methylation there is, actually. We found actually some of these TEs are kind of in, no, they're not in promoters, they're actually in transcribed regions. And that's the difference between mammals and insects, is that it doesn't tend to happen in promoter regions.
Okay. And so I just want to, I think I can't remember whether it got more when the treatment, the insecticide, did it remove methylation or stimulate methylation? And those levels on that x-axis, what was the x-axis? That would seem, what was the values on the x-axis? Like I saw 0.03, 0.0.
Yeah.
That's small. What
Yeah.
was the unit?
That is total CPG methylation for the genome. For the
That was global
global,
average-wise?
yes.
I don't even know how you detect that low, but.
Yeah, it's very low overall.
Yeah,
And so
okay.
maybe, that's the one thing to take, with a grain of salt, is we don't know how much, these minuscule changes actually varies. So really kind of with insecticide exposure, that actually led to hypomethylation.
That's what I was.
Yeah, yeah, yeah.
I'm also interested about the T methylation. So because you see there's an association between methylation and T, do you also have a chance to test whether T actually expand across the genome? You have more copy of T or have less copy of T, less number of T? The other associated question is that do you have any idea about what causing demethylation to increase or decrease? Is it because direction from the T to actually regulate methylation, or is it because methylation actually regulates T expansion or shrinkage?
Yeah, so in the literature, not in our own work, like there is a lot about methylation regulates kind of T activity. And then in terms of our work, we've done TE work in looking at kind of like sort of a population genomic study approach. But honestly, because the background of these insects was so variable, we did not decide to look at sort of this proliferation of TEs because we were unable to follow. We didn't do kind of a whole characteristic of parentage, so we couldn't do that. So now we are planning to do it with this upcoming data set because we know exactly all the parents, and so we can look at this idea of following exposure to insecticides. Do we see TE proliferation? So we know that there's about 330 active TEs, and so we've focused on their mobilization. And we know from individuals, it's something like 8,800 unique locations across every single insect that we've looked at TE mobilization, that there's a lot of activity all the time. And so the initial kind of estimate of the genome size for the beetle was 450 megabases. Now it's 1 gig because of this. There's a lot of this dark matter kind of thing, that sort of unsequenced areas, probably because of active TE, movement. Okay.
Thank you very much.
Morning, everyone. My name is Maro from Fudan University, and today I'm going to talk about the pyroclastic response and adaptive potential to project salinization in freshwater fish. And for the background, As we know, freshwater ecosystem is very important because it provides habitat for at least 9% of the described animals in the world. However, due to the seawater intrusion and other reasons, the salinity in freshwater ecosystem is increasing around the world. And the salinization have the negative effect on the freshwater organisms. For example, the salinization can cause the osmotic stress in freshwater organisms and reduce the growth and influence their reproductive success. Even can pose a threat to the biodiversity of the freshwater organism. And as the main vertebrate of the freshwater ecosystem, how do freshwater respond to salinization? There are three ways: migration, phenotypic plasticity, and adaptive evolution. However, due to the high fragment of freshwater ecosystem, migration is very difficult for fish, so they will respond to salinization by phenotypic plasticity and adaptive evolution. In our study, we will focus on phenotypic plasticity and for the phenotypic plasticity, most studies have focused on the behavioral, physiological, and developmental plasticity under salinity stress. But the underlying molecular mechanisms have been less explored. And last study investigate the difference or similarity of the plasticity between freshwater fish with different history of previous short exploration. And the underlying molecular mechanism, microRNA is a very important role in the freshwater organisms to regulate their plasticity in response to environment change. And so, based on the background, we want to know what is the similarity or difference in plasticity between freshwater fish populations that have experienced different salinity environments, and what is the molecular mechanism underlying the plasticity response to salinity stress in freshwater fish. And in our studies, we want to focus on the gene expression because gene expression is the process of plasticity. And we also focus on the microRNA, the important epigenetic mechanism. Okay, to answer our question, we collect two populations, one from freshwater stable environment and another one is from salinity fluctuating environment. And we choose the western mosquito fish as the study system because these fish may live in the freshwater, but they have broad salinity tolerance range, and it is suitable for us to study our question. And we collect these two populations, and after acclimation for at least 4 weeks, we readily assign these fish to freshwater or the salinity stress, and we want to compare their plasticity for at least for 1 hour, 24 hours, and 48 hours. At the end of each time point, we perform and we collect the gill tissues, and we perform the RNA sequencing and small RNA sequencing. And for the result, because we collect the two populations from different environments, so we want to know their population structure, and we found significant genetic structure between the two populations. And we want to know the factors influencing the global gene expression between of these fish. So we analyzed the factors, and we found microRNA expression variation and population structures is with the highest proportion of the global gene expression variation. And then, because we also want to know the similarity or difference between their gene expression, so we first identify their salinity responsive genes in each population, and we then identify their shared salinity responsive genes to analyze their expression direction between freshwater and the salinity treatments. And we found there are most of the salinity responsive genes shared in both populations, and they show significant tendency for parallel expression change across time. And we also analyzed their nucleotide diversity, and we found the parallel salinity responsive genes showed significantly higher nucleotide biodiversity. And this result suggests that although they have experience in different environments, they will use the same genes responding to the salinity stress. And we also want to know whether the microRNA involved in the regulating of the gene expression, and we analyzed their role. And we found that the parallel salinity responsive genes were regulated by the microRNA, but in each population, there is a small overlap between the population. Okay, to summary. And we found a course of genes with parallel expression change in both populations, and these genes were involved in osmoregulation. And also, we found the shared salinity responsive gene with the same expression direction showed greater nucleotide diversity than other genes in the genome. Also, we also found that the parallel salinity responsive genes were regulated by different microRNA in each population. Okay, that's all of my presentation, and I want to thank the co-authors, Jintao Hu from Fudan University and Rowan Barra from McGill University. Okay.
So when you said that there was greater diversity in terms of the gene expression, was this in kind of all the same gene families, or were they kind of really different gene families?
Oh, this is a good question, but we also want to further to analyze this. Yeah, we haven't done this, analyzed this. Okay.
Do you have any speculation why you think the gene expression change is the same, but the microRNAs that are correlated with that are different in the two populations? You've got the same gene expression, but different microRNAs that are correlated to that.
Yeah, we found the same gene involved in the organ regulation, but they were regulated by different microRNA. Very small overlap between of the two populations.
Yeah. How do you think that came about?
Okay, maybe we can communicate later. Thank you.
Do you think there might be a correlation between some genes that are making the microRNAs and the microRNAs that are structuring the genetic variation that could explain the microRNA differences?
Please. Say it again.
So a correlation between genetic variation that are making the different microRNAs?
Junior? Okay, we can talk.
All right, I'm going to tell you about how host plasticity and the microbiome underlie acquired salt tolerance in a freshwater plant. So I'm going to start off with reminding us that we understand genetic adaptation fairly well, obviously not well enough because we still got a whole conference all about it. But we know that allelic variation in the genome can impact trait values, which can then impact fitness. And as a result, then that impacts the frequency of these alleles in the next generation, leading to adaptation and evolution. But there are also, we're also hearing a lot about adaptive changes to phenotype that don't exactly exist in the genetic code, so epigenetic modifications, such as methylated cytosines. In some organisms, these heritable, or these epigenetic modifications are actually very easily inherited, for example, in plants that reproduce clonally and vegetatively. We can also have other extra genomic effects on traits. So when species interact with each other, that can result in trait change. And here I've drawn up a little example where there's a second species that has variation in its genome for how it impacts the trait of that green species. And if that trait or the species of the fitness of the green species feeds back to affect the fitness in the purple one in this diagram, then we'll get evolution of the trait in the green species and the purple species genome. So this kind of evolution probably happens on the regular in very closely interacting pairs of species, such as, for example, microbes that live in close association with hosts. Okay, so the study system that I'm going to tell you about today, in which we're trying to disentangle genetic, epigenetic, and microbial contributions to adaptive trait change, is duckweed, Lemna minor. It's a cool, and I'll explain those asterisks later. It's a really cool plant for this study because it's really small. It fits in a 96-well plate, as you can see there. It's got a short lifespan, so depending how good the conditions are, it might be as short as 3 days. And we can manipulate all those cute little microbes by bleaching the fronds to get rid of them and putting them back or not putting them back. We've also got this camera system that runs underneath 96-well plates, takes pictures, and a computer software that turns those pictures into data on the amount of duckweed frond area or fitness over time. So the model selective agent today is going to be salt and fresh water, so thank you for that introduction. This is a really important change to our environment. In temperate areas like this one, winter road salt can exceed 30 tons per mile per year, which is making our freshwater increasingly salty. In temperate North America, in Toronto, which is where all the collections I'll talk about today come from, that salt level increase of salinity even persists into the summer in freshwater. So duckweeds, we learned before we started this study that duckweeds in the area didn't have much genetic variation, but yet they're a freshwater organism living in sites that get pretty salty. So we thought maybe there's something not genetic going on in their ability to tolerate this. And so all the experiments that I'm going to talk to you about have a similar kind of treatments. And this is the design of the first one, where I'm manipulating one epigenetic mechanism, here the methylation of cytosines, so that 400 micromolar zebularine treatment is knocking down the amount of cytosine methylation. We're also manipulating those microbes, so this is all of the plants had the microbes bleached off of them, and then the plus microbe treatments, we put back microbes from their home site onto the plants. And we expose them to different salt conditions, with 0 being the appropriate freshwater condition, 5 grams per liter representing a runoff event, and then 9 grams per liter representing a fairly severe road runoff event. There's also variation in what duckweed line we're including in this first experiment. So that little figure, cryptic figure on the bottom is a map. The big black blob is Lake Ontario, and then you'll see sort of a little grayish cluster next to the end of that, and that's because I've drawn all of the roads in the area on that map. Each red dot is a place where we collected duckweed and isolated a single clonal line, and you can see that some of those clonal lines are coming from close to that cluster of gray lines, which is Toronto, the city of Toronto, and some of them from farther away. So they differ in their road density and the extent to which they're experiencing road runoff of salt. Okay, so I've just put up all the data on the screen, but don't panic, I don't actually want you to see too much in this plot. On the x-axis of every little subplot is the days in the experiment, so around about 12 days. On the y-axis is that total frond area, and then each of those lines is the average of one of those clonal duckweed genotypes. And then we've got in the columns, the different salinity treatments, and in the rows, the different microbe and methylation treatments. And all I want you to see here is that if you go increase in the direction of higher salt, the plants are growing less. That's it. For everything else, I'm going to zoom in and make a much simpler metric. So I'm going to tell you now that the color of the line means something, and that's to do with road density, with the dark black lines indicating urban sites and the green lines indicating rural sites. And the simple metric, which I'm just going to have to assure you, reflects the model predictions for all of the data set. But the simple metric is we're just going to take the average growth rate of the most urban line, the average growth rate of the most urban line, and subtract the two, and then do that for every condition. So it's generally positive, so I'm just going to call it the urban growth rate advantage. And now it's on the y-axis here. And so what we're first looking at here is just all the, quote, normal condition treatments, where I've given them back their microbes and I haven't messed with their methylation. And in these conditions, the urban lines are generally growing faster than the rural lines across the salinity treatments, but especially that mid-range salt near the average salt runoff event. If I take away the microbes, that's gone. So what that means is that the microbes are definitely contributing to the ability of duckweeds in urban sites to tolerate high salt levels. If I leave them with their microbes, but I reduce cytosine methylation, it's also gone. So that means that also cytosine methylation is important for that urban salt tolerance. If I take away both of these things at once, we recover just a little bit of the trend, and now the variance is reduced. So there's a little bit of an advantage for the urban lines growing in that medium salt condition. What that tells us is that there's additional factors at play, which probably shouldn't be surprising because they do have genomes that can change, and they also have some other ways that they could be regulating their gene expression other than cytosine methylation. Okay, so before I launch into the next experiment, you're just going to look at a picture of microbes on a duckweed root for a while to clear the palate. All right, so this is a smaller and larger experiment. The containers got bigger, and the number of different treatments got smaller. So fewer salt treatments and just one duckweed clonal genotype. But now we're in a big container, so we can take samples for microbial metagenomics and methyl-seq, and we're going to run it for a long time. And this experiment is therefore sort of a salt press experiment. And the really easiest result to see here is that microbes increase salt tolerance, but only because microbes are just increasing growth across all treatments. So we've got the micro treatments in the yellow circles, and if you compare them to the other treatment in that box, you can see, wow, more fronds inside the yellow circle. And those are just random jars from the treatments. Okay, but we're really interested in what's going on with salt here, and so now we're looking at the effect of 5 grams per liter salt on the frond area, so the 5 grams per liter jars compared to the not salty jars. And so when it's peaking at that negative, or at the minimum, at about 12 days, that means that the duckweed in the salt jars are not doing so well in comparison to the duckweed in the unsalted jars. However, by the end of the experiment, salted and unsalted jars, same. So the salted jars, the duckweed recovered and ended up growing to the same amount by the end of the 35 days. Okay, we were hoping in our methylation blocking experiment that we would see, hey, reducing this methylation is important to their ability to recover. We thought it might block their ability to recover. However, it blocked the negative effect of salt in the first place. So we really don't know at this point in the talk whether or not that methylation was important to the recovery from salt stress. Okay, I also told you that there would be microbiome sequencing, and it turns out that if you have a lot of microbiome metagenomic data, there's a lot of predicted gene functions and taxa that differ across your treatments. So I'm just going to show you some quick highlights that are the things that I think are maybe the most obvious to look at, and that is looking at the two most abundant taxa, looking at the two genera and the two functions that explain the most variation among sampled communities across the samples using a PCA. And all of these three results are going to look very similar, where I've got a biplot showing all of the samples, and then a single plots on either side showing you the treatment means. And so just to quickly summarize, microbiomes in jars without salt have more Rhodococcus and fewer Pseudomonas, which are the two most abundant genera. They also have fewer Parvobaculum and more Colobacter, which are the two genera that explain the most variation in community composition. And they also have more predicted chlorophyll genes. Both of the functions that were most correlated with variation in communities, or that explain the most variation in communities, were chlorophyll genes. So there's chlorophyll gene on both axes. Predicted genes, right? So we're just looking at predicted function. Okay, I also promised you methylation-seq data, and I do not have it. So, false promises. And the reason is I'm going to now explain those asterisks from the beginning. So I told you I had lemna minor, but what I've actually got is a triploid hybrid between lemna minor and lemna trianifera, and we ended up having to go the route of de novo genome assembly just to get our reads to map. So we are still at the mapping the reads stage, despite the amount of time that has elapsed between the sequencing and now. What we'd like to do with that and the microbiome data is to compare differentially methylated regions between our 0 grams per liter treatment, our 5 grams per liter treatment, and then also look at our original duckweed lines, those 10 ones from the earlier experiment, and see if there's any regions that are differentially methylated in the experiment and also in the real sites. So that's to come. And then this is another pause slide. And I want to then quickly follow this up. So we've got now a follow-up experiment to this jar. So we can take duckweeds and microbes out of these jars that we know the exposure history to salt from, and mix and match now duckweeds from those no microbe jars with different salt history with microbes from different salt histories in new salt exposures. So that's what we're looking at now on this slide, where we're looking at the fitness of duckweeds from different salt histories in either their condition they just experienced for 35 days or a different condition. So remember, these are clonal lines, and 35 days is about 5 duckweed generations, so we don't expect this to be evolutionary change, we expect this to be acquired plastic change. So here's where we recover from that zebularine issue. So we see this sort of local adaptation to recent salt history in some sort of plastic way. If I add microbes, I'm just going to add them both at once, we see a reduction in that pattern. So, and it's especially bad if we add microbes that experience the same salt history as the duckweeds. So that's sort of covering up this trend of local adaptation in opposition to the first result. So to summarize, duckweed microbes can contain evolved salt tolerance, reduce the need for it, or obscure it. So all sorts of things. And I just want to quickly plug that my lab is new. We've got some cool eco-evo microbiome stuff happening, and projects hopefully coming soon in, for example, microbiome-derived trait plasticity. With that, I'm going to wrap up if there's time for a question. Yeah.
Just have a quick question about the grocery difference between the duckweed with the demethylation agents and the salt. So because you are adding microbes to the experiments, so are you assuming there's a direct causation between the demethylation agents and the duckweed itself, or it's because the demethylation agents actually it affects the microbes and then affects the duckweed? So
Yeah.
Yeah,
we wondered about that. I think it's probably affecting both of them, right? Just because anything you add always affects microbes, no matter what. Most of the microbial methylation is probably not cytosine methylation, so it's going to be affecting them in a different way. But I'm going to go ahead and unlock a bonus slide. And that's, there's, in the special case where we add microbes and this demethylation agent, we get weird deformed fronds. So there's definitely some impact of this agent that depends on the microbiome. So, yes. Yeah. Yeah. Go ahead. Sure, yes.
So I have two questions that are related. So how do you get rid of the microbes, and how do you reinsert the microbes? And could that manipulation also not affect your results?
Yeah, so we remove the microbes. So long before the experiment starts, we take our clonal line and we bleach them. And then we bleach them again and we bleach them again until we don't get anything growing in a high nutrient media for a while. And then we say, well, most of the microbes are gone. And so that's how we get rid of them. How we put them back is we took swabs of plants from the field and we cultured them. And so the microbes that are going back on are going to be some sort of interesting subset that can grow on a plate from the field, because we needed those microbes to wait for us to bleach all of the other microbes off the plant. And you had a two-part question, and I totally forgot.
So could that affect your results? Or
Yeah, I think.
Is there a way to control a bit better these?
I think it's likely to undercount the contributions of the microbiome just because we're missing so many taxa. That's it. Thank you.
Thank you. Hi, everyone. So yeah, I'm going to talk to you about DNA methylation and climate adaptation in trees. So, as I'm sure everyone knows, plants show massive phenotypic diversity in response to environmental change. This figure on the right is a map of California, which is shaded by the precipitation gradient. And the two photos show the same tree, Valley Oak, which is what I'm going to be talking to you about today, occupying very different ecosystems along that massive climate gradient. And thinking about climate change, trees are a specific concern because they live for a long time, some for several centuries, and so they may be lag adapted to the environment when they germinated. And we're seeing examples now from oaks and pine of this. And so studies are beginning to focus on the genetic reasons for this maladaptation in trees. But mostly, the epigenetic contribution to this is overlooked. And actually, if we can understand how epigenetic mechanisms, epigenetic being, well, we all know we're in a session. But yeah, so we could really start to unpick both genetic and epigenetic difference to this adaptation. So here we focus on DNA methylation because it's the best studied epigenetic pathway in plants. And we already know that DNA methylation is important in plant developmental processes and in the regulation of gene expression. So here we have a kind of cartoon of the three contexts, and you'll see that CG and CHG are symmetric, and so in gene transcription they can be exactly copied. CHH on the left, so H being any nucleotide, or being A, T, or C. There's no corresponding cytosine nucleotide on the other strand, and so it's asymmetric, cannot be copied in gene transcription, and so is therefore not heritable. And historically, DNA methylation was viewed as a silencing mark. So in this cartoon at the bottom, you can see methylation either upstream of the gene, so maybe in the promoter region, or in the gene body, was thought to silence. But new evidence is showing that actually DNA methylation, either in the promoter or in the gene body, can be associated, positively associated with expression. And as a shameless plug, we discussed lots of these ideas in this Trends in Plant Science opinion paper, which came out very recently. So my overarching question here is, does DNA methylation play a role in phenotypic diversity? So can methylation explain either the drought-stressed Californian oak in the top picture or the thriving Californian oak in the bottom picture? And so ultimately I'm trying to use natural populations of trees to investigate what effect epigenetic pathways, DNA methylation, have on evolution. So I'm going to start with a pilot study from my lab group, which is unpublished. But the same Valley Oak trees were sampled in autumn and spring, and they're all growing in a provenance study. And we sequenced reduced representation methylones to look at the effect of environmental plasticity. So does methylation change between season? And so this bar plot is just showing the average methylation of the significant variance.
And you can see for CG and CHG, there's no difference. But for CHH, we have a big reduction in spring. And so this led us to question, could this environmentally induced CHH methylation explain these different drought phenotypes that we see in oak? So I'm going to use two studies to answer two questions, all in Quercus lobata, which is valley oak. So the first one is a methylome study. So is DNA methylation associated with gene expression in oaks? And the second one, I'm going to use experimental data where we drought-stressed oak seedlings to look at, is DNA methylation induced by that stress? So for the methylome study, we sampled 42 valley oak trees growing in a provenance study, which is the purple star on that map. The map of California is shaded by maximum summer temperature. And these trees make up maternal genotypes from all across the species range, so those are the black dots. And we sequence whole genome methylomes and transcriptomes at the same time to try and unpick any interaction. And so this is the methylation data. So these are metaplots showing the average methylation across genes. So you can see I've annotated the gene body on the x-axis, and you've got the three contexts, so which all look very different. So CG, we see this peak in the gene body. CHG, we see no methylation in the gene body. And CHH, we have this peak upstream in the promoter region. And so if I overlay this with the expression data, and so this is the same plot, but I've just taken the top 20% most expressed genes and the bottom 20% least expressed genes to compare. And you can see there are some stark differences. So in CG, we have a bigger peak in gene body methylation in the most expressed genes, and absolutely none, or nearly none, in the least expressed. And for CHH, we see a big peak at the promoter in the most expressed genes. And so we can see that these two contexts are correlated with expression. So yes, to our first question, CG and CHH are correlated. So then to look at the second question and second study. So for the experimental study, we collected individuals from just one site this time because we wanted to reduce the amount of genetic variation that we would get. So we took 8 genotypes, took them to UCLA and set up a greenhouse drought experiment, and then analysed differential methylation between the samples. So this plot is showing the average transpiration rate in the drought-stressed individuals minus the average transpiration rate in the control. So you can see 0 is the top on the y-axis. So it's all below 0. The pink bars are the 2 periods of drought stress. So we did a short 10-day drought to prime the plants, and then a longer 20-day drought. And we recovered the plants in between. So you can see the line kind of nearly gets back up to 0. And I sampled the plants at these 4 time points. So before the drought, at the end of the priming drought, where you can see there's very little phenotypic difference. Then midway through the longer drought, where the leaves are starting to look a bit darker. And then at the very end, where the leaves are mostly dead or dying. And so I sequenced 2 genotypes, this is just a pilot study, from these 2 time points, and we sequenced Oxford nanopore long-read genomes and methylomes. Okay, so then the data from this. So this plot is showing the average differentially methylated positions, which I call DMPs. I know that's different to all the other talks we've had, but so the average number of differentially methylated positions in the 3 contexts along the x-axis. And the drought-treated individuals are shown in orange. So if we start with CG, there's no impact of drought on CG methylation. But if we look at CHG and CHH, you can see that there's lots of differentially methylated positions following drought stress. So next I wanted to look at what's the impact of this, because differentially methylated just means it's different. And so I wanted to see, is it more or is it less? So this is the same plot, but now split into, so the top plot is hypermethylation, so high methylation, and the bottom plot is hypo, so removing methylation. And so you can see for CHH, it's basically all hypermethylation, so drought is inducing lots of methylation. Whereas for CHG, you can see that the drought-induced methylation is pretty much 50-50 split between addition and removal. And so then I wanted to see what is being methylated in the genome of these drought-induced differentially methylated positions. And so this plot is just showing the drought DMPs, but now is shaded by whether the methylation is over a gene body, a promoter region, or a transposable element. And so again, for CHG, you can see it's an even split between hyper and hypo, and between gene bodies and transposable elements. But for CHH, we have this massive hypermethylation almost entirely of transposable elements. And so to show that in a slightly different way, this is a metaplot of the transposable elements which have differential methylation on top of them. So time point 1 is the samples from before the drought stress, so I'm comparing the same individual plants here. And then time point 3 is midway through the drought stress. So you can see that there's a big increase in methylation over these transposable elements. And just for comparison, the right plot is showing all the other TEs in the genome which don't have differential methylation over them. And then the final thing is I wanted to look at gene expression. So we don't have RNA data yet from this experiment, but a previous experiment in my group, which was also looking at drought stress in Valley Oak, has like 2,500 differentially expressed genes. So I took them and I looked at the methylation of those genes in this experiment. And you can see that in CG context, so the drought, it's the same colors as before, so blue is the control and orange is the drought. And so you can see that for CG, we see an overall reduction in methylation over these differentially expressed genes. No difference in CHG. And in CHH, you can see, again, a small peak both upstream and downstream of the gene body in these drought-stressed individuals. So back to our question. Yes, we do see that DNA methylation in CHH is induced by drought stress. And so to conclude, so we see that CHH is higher in drought-stressed seedlings. It targets transposable elements and upregulated genes. But as I said at the start, it's not heritable. And so one line of thinking in trees is that because they are these very long-lived organisms, maybe DNA methylation is more important as a way to flexibly change the expression of genes. But we think this is evidence of DNA methylation as a potential source of climate adaptation in oaks. And so I'd like to thank all these people, Ryan who made the maps, and yeah, thank you all for listening. So our original theory was that it's TEs in promoter regions or in genes which are being methylated to kind of silence the TE and switch on like gene expression. But in this figure, I took all of the TEs here, which are being methylated, and then looked to see where they are in the genome. And only about 20% of them are in a promoter region. I mean, maybe that's high. But yeah, the majority of those TEs are not on top of any kind of gene region. But I know in one of the earlier talks you looked at within 2 kb of a gene. So I could also do that and see. But yeah, we don't really know, is basically the short answer. TBC.
Drought stress to increase TE activity, and that's why CHH are there to control them from jumping around?
Yeah, so that's why we sequenced like long-read genomes, is to see if drought induces structural variants, and like, yeah, structural variants being like mostly TE insertions. So that data is still coming. But that's definitely what we're wondering.
Just had a random idea whether you see kind of any natural variation in sulfur availability, you know, because say like with the one carbon cycle, it's dependent on sulfur as being sometimes a limiting factor, and that would be kind of an interesting thing just to check on the side.
Yeah,
whether that
interesting.
influences,
Definitely
like,
something to look at.
the response.
Yeah, thank you.
Okay, thank you very